Okay, so in Google Classroom, in the classwork tab, okay, and it'll be under labs, so a little bit further down, cell anatomy lab. Okay, obviously you want the Google Doc open. I'll be working out of the PDF, and then when I get done explaining all of this, then we'll have the recording in there as well. Okay, so your Google Doc looks like this. Okay, you're just going to fill it out, same as we always do. All right, so the problem I have written here is that cells have to carry out the basic functions of an organism in order to survive. Okay, thus they have to have structures within their cells that carry out these basic functions. What are you supposed to do with the problem? Write it in your own words in the form of a question. Okay, so you've got to figure out something there, okay, that kind of asks, what organelles might be necessary, okay, for it to do its job, something like that. Okay, figure out how to put it in the form of a question in your own words. Okay, so the whole purpose of this lab is to see if there are common structures between all of the cells we're going to view. The idea we're working on here has to do with the second point of the cell theory. It's in the problem. Right? Cells have to carry out the basic functions of an organism. Okay? What do they have to have in them in order for that to take place? So we're going to look at a plant cell. We're going to look at some animal cells, okay, tomorrow. And we're going to see what do they have in common. It would be logical to assume that any structures that are in common are pretty important. Would that be fair? Okay. Now, is it possible there could be some structures that are not common to both that are also still important? Okay, but those would have to do with their what? Partly, I'm thinking of three words here. M-O-N. Modes of nutrition. Nice. Well done. Okay. So if there's anything that's not in common between those cells, and there's going to be, and some of them you'll actually be able to see, it would have to do with their mode of nutrition. Okay? Anything they do have in common is obviously helping to carry out some of the basic functions of the organism. Okay? So if we're looking at our variables here, okay, in this experiment, we're looking at cells from different types of organisms. In fact, what we're kind of doing is repeating Schleiden and Schwann's experiment. We're looking at cells from lots of different organisms. We're trying to see what structures they would have. Okay? We're looking for common structures that carry out basic functions. What are some things that we would need to control each time we look through the microscope? I'll give you one of the controlled variables right now. In order for this experiment to work, all the cells we look at have to be eukaryotic. Okay, I talked about that the day that I went over the parts of the cell, okay, and what they do. We are a eukaryotic organism. Our cells have organelles. Okay? Prokaryotic organisms are single-celled organisms, and their cells don't have organelles. Okay? So this experiment would not work if we looked at prokaryotic cells. Okay? So one of your controlled variables is that all the cells you look at are eukaryotic. Okay? I think you can probably come up with two more. Remember, you need to explain all 5 variables, manipulated, responding, and all 3 controlled. Okay? So for the manipulated variable, you're explaining how and why it's being manipulated. For the responding variable, okay, what you're looking for, how that's responding, why it's important. For your controlled variables, how they're being controlled, and the effect they would have if they weren't. Okay, for our hypothesis, what's the if part of your hypothesis always supposed to talk about? The problem. Yeah, so I would say the if part of your hypothesis should probably talk about the second point of the cell theory. The and part, as always, just describes the experiment. Eukaryotic cells are viewed under a microscope at high power, looking for various structures, whatever, however you want to say it. I mean, we're looking at cells under a microscope, looking for stuff. Don't say it that way, okay? But that's the gist of it. Okay, and for the then part, you should probably predict what structures are going to be common or visible. And why? Now you have to be realistic. Okay? If you're telling me that, you know, ribosomes are going to be visible under the microscope, that's not a hypothesis you're going to be able to accept later. Okay? Ribosomes weren't discovered until the advent of the electron microscope, so you ain't seeing them. Okay? Not with what we've got. But you should be able to see some of the more obvious, larger ones. Okay? And I'll show you some pictures of what we're going to see tomorrow, just so you get an idea of what we're going to be looking at. Okay, so the three things that you have to get pictures of tomorrow through the microscope are going to be the monocot dicot cross section. That's a plant cell. Okay, when we look at that, okay, this is actually what it'll look like on high power, but okay, your slide for the plant cell will look like this. And it'll have these two like wedges in it. Okay? The top one is a monocot, and the bottom one is a dicot. You don't need to know what that means. It's different types of plants. You want to look at the bottom. Okay? So you want to look at the dicot one. Right? So make sure when you're on scanning power, okay, you move the stage around so you're sure, okay, I'm on the bottom one. Okay? So I'm looking at the right stuff because the structure of the leaf in a monocot is different. Okay? And it won't look like what we've been showing. Okay? So that's the first thing you're going to look at. When you look at that under the microscope on high power, okay, you will see this. Okay? So here's your epidermis up here at the top. Okay? The waxy cuticle is kind of invisible, so you don't really see it. Okay? And then here's your palisade layer. Okay, see all these kind of rectangular cells here? All right, you see all these green ovals? Each one of those is a chloroplast. Chloroplasts are quite big. Okay, you will see them under the microscope.
Do we have to label these?
Yes, you will have to label these. Okay. All right, here you got a nucleus. Okay, and if you look really closely at the nucleus, you can see there's a nucleolus inside of it. Okay, and if you look over here, kind of on the top of this one, okay, right in here, okay, you can see that it almost looks like the, well, if it was a face and it was upside down, it's almost like the nucleus has a beard. Okay, here on the end, that's the rough ER. Remember, there's always rough ER stuck on one side of the nucleus, and you can usually see that with the microscope as well. You have to know what you're looking for, which is why I'm kind of illustrating it right now, so you know what to look for. Otherwise, you kind of get lost quickly. Okay? Now, can you see cytoplasm? Well, I see a lot of empty space within the cell, and that's what cytoplasm kind of is. So yeah, I could label cytoplasm. Okay? Can I kind of see where one cell ends and the next one begins? Then I can kind of see where a cell wall or a cell membrane would be. Okay? I'm not saying use your imagination, but use your imagination a little bit. Okay? I'm not saying guess, but you know, be logical about what you think you're seeing. Okay? Again, you're not going to see ribosomes. Don't tell me that. Okay? But you can see a fair amount of stuff. Okay? Within here. So, you know, go for it. Label it. Okay? I'm not expecting you're going to see everything that's on your cell diagram. You're not, and that's okay. But you can see some of it. Some of it. Okay? Like even in this one here, right? Right there is a Golgi apparatus. Okay? Probably hard to see for you guys, but you can see a fair amount of detail on this. Okay? Now, what you might also want to do is if you can get a really good picture with your phone, blow it up. Okay? I mean, you can see fairly well magnified through the lens, but if you take a picture of that and then magnify it a little bit more, it might even be easier for you to see that level of detail because you can magnify again after you take the picture. All right, so that's the plant cell. Okay, the next thing we're going to look at is the generalized animal cell. Now, when you look at the generalized animal cell, what you're really looking at is something called stratified squamous epithelium. If you've ever dusted your knuckle, it's that really red skin underneath. It doesn't bleed a whole lot, but stings like crazy. Okay, that's that stuff. It's the underlayer of your epidermis. Okay, and so when you get that under the microscope, okay, this is what you should be able to see. Right? So can you see the individual cells up in this part here? Right? So if I'm doing a half decent job here of kind of picking a cell, this would be one cell. Okay? So there's a whole bunch of them. Right? I always tell kids it looks kind of like dried mud. Okay? And when a mud puddle dries up, it all cracks. Okay? It kind of looks like that under the microscope. Okay? Obviously, we got a nucleus in the middle of that. Okay? And we can pretty clearly see where the membrane of that cell is. Okay? Also, on the left-hand side of that nucleus is some rough ER. Okay? Nucleolus is a little tougher to see in that one, but I can certainly see it in this one. Okay? There's a little dot in the middle of that. Okay? If I blow it up even more, it'll become even more obvious. Okay? So yeah, I can see a nucleus pretty clearly in there. Okay? I would say that's probably a Golgi apparatus, that little dot right there. Okay? Probably a Golgi there and there. Okay? Well, sorry, right there. Not sure what the other little dot would be. Okay? But again, I'm not guessing. I'm just being logical about what I'm pointing out. Okay, I can see a cell membrane. If I can see the nucleus, I can see the nuclear membrane because I can see the outside of it. Okay, these are all things that are possible for us to label. Okay, now can I see lysosomes? Possibly. There's a lot of kind of grainy stuff inside this animal cell. Okay, one of them could be a lysosome. Right, so I could certainly. That's probably something I could label if I thought I saw one. Okay, everybody all right with that idea? Okay, so that's what you're going to be looking at, okay, for the generalized animal cell tomorrow. Okay, the third thing you're going to do is, oh, maybe we can't do this. I never thought about it. No, we definitely can't do this. I can't have you making a swab of your own cheek during COVID. Okay, I will post the pictures of mine. Cheek cells instead. I will put them in there because we obviously can't do that. But I'll put some human blood out there. You can look at that. You don't have to take a picture of it, but everybody always asks. Okay, but I'll put some human blood out there. But okay, for your own cheek cells, okay, these are mine here. Okay, so that's just a cell taken from the inside of my cheek with a Q-tip. Okay, so I can see a nucleus pretty clearly. There's some rough ER there. Okay, you can see the membrane going around the outside, nuclear membrane, nucleolus. Okay, this is probably over here, the Golgi. These would probably be lysosomes. Okay, notice I don't see any smooth ER. Smooth ER is nearly invisible. You won't see it. Okay, most likely because it doesn't have anything to mark its presence. But these are harder to see because they're not stained. This is stained. Okay, they put a dye in it so everything stands out. Okay, but your cheek cell will be a little bit tougher to see in so much as it's not stained. Okay, this would be a few other cheek cells. Okay, got a bunch of them on there. Okay, nucleus, rough ER, probably right there. That's probably Golgi. Okay, lysosome, cell membrane, nuclear membrane. Right. So I'll put those pictures both in the assignment. You can label those. Okay, so unfortunately you won't be able to look at your own, but I don't think that would be a terribly responsible thing for me to do. Luckily I thought of that today and not tomorrow after. Okay, yeah, and I'll put some human blood on there. This is what human blood looks like. Okay, there's lots of different kinds of, there's lots of red blood cells, but there's also leukocytes. Okay, those are white blood cells. And you can see those because people always ask. And no, you can't look at your own, obviously. Okay, so don't do that. All right, so the labeled diagrams that I'm going to want for this lab are going to be the plant cell, the animal cell, and the cheek cell. Okay, so those three things need to have lab diagrams that are labeled. okay? In the same way that you're doing it for your microscopy lab tomorrow. Okay. Now, so that's all the procedure stuff. Okay, the analysis, there's only one analysis question. But I'm expecting a paragraph answer here. Okay? So based on your knowledge of cell structure and your observations of different cells in this lab, what organelles are most important for a cell to function? Okay, so the first part of your answer has got to be, when viewing through the microscope, these were the structures that were common to all the cells. Okay, this is what they do, this is why they're important, so kind of reviewing what you know about the structure of a cell. Okay, the second part, okay, and which ones were specific to a certain mode of nutrition. All right, well, in the plant cell, we saw these things. They weren't present in either of the two animal cells. They carry this out. That's, photoautotrophs. Okay, talk about that. Same with the animal cell. Okay, explain your reasoning. I'm looking for probably 4 or 5 sentences worth of explanation here. Okay, it's only one analysis question, but I'm looking for you to tell me what you know about cells, basic functions, and what organelles are responsible for. Okay. Then your conclusion, copy paste your hypothesis. Did you see the things you said you were going to see? Why did you see them? Why didn't you see them? Accept or reject. Okay, and then your sources of error. Okay, so 2 sources of error and ways to fix them. Questions on what we're looking for here? All right, so you still got around 20 minutes, okay, to get all the pre-lab stuff done. If you get the pre-lab stuff done, then you got a little time to work on your microscopy lab that's due tomorrow. Okay, you got some time here with the Chromebooks. Make good use of it.